Okay, could somebody confirm you can hear me?
Yes.
Yep.
Thank you. All right, before we begin, are there any questions? All right, if there's no questions, I have done some grading. I have most people's lab 2 graded, and I hope to finish the rest of the people's lab 2 tonight, probably during the lab session tonight. Um, I'll talk more about the lab in the lab. And I also got the worksheet for the previous lecture graded. Are there any questions on the grading or anything like that? All right. We had talked about phospholipids last time, and we mentioned that they are a biphasic molecule with the phosphate portion of the phospholipid being uh, hydrophilic, meaning it likes to dissolve in water. And then the tail portion of the phospholipid, the fatty acid portion, is hydrophobic and it likes to be away from water. Phospholipids are the main molecule in the cell membrane. When you get a bunch of phospholipid molecules and you put them together in water, they will start lining up like this. Let me make this a slideshow. Well, they'll start lining up like this so that the fatty acid portions of the molecule will come together and be away from water, which is on the outside. And then the fatty acid portions will be together and the heads will face the water and it'll just form a circle like that with the water being outside and the heads being on the outside of the phospholipids, the fatty acid tails all being on the inside. And then there will be a layer of phospholipids with the head facing outward. And the inside of this would be, of course, water inside here. And this should remind you of something. What does that look like to you? Anyone?
Sunflower or lemon?
Okay, let's go to microbiology. This should remind you of a cell. The cell will, especially an animal cell, will be surrounded by the cell membrane. And that's what this is here, with phospholipids being on the outside of the membrane, and I mean the phosphate head being on the outside of the membrane, and the phosphate heads being on the inside of the membrane, with the tails making the phospholipid bilayer of the cell membrane. And then the outside of the cell, if we're talking about in blood, will be mostly water. And it doesn't really matter where the cell is because most of the body is water. So wherever the cell is, mostly it's going to be water outside the cell. And then mostly water inside the cell. And that will be the cytoplasm inside the cell. So this should remind you of a cell. And as I stated, the cell membrane is mostly composed of phospholipids, with the phosphate portion being the outside and on the inside, and then the fatty acid portions being inside the cell membrane. All right, any question about that? If not, let's talk about another group of lipids that we call steroids. If you remember, there were three types of lipids: simple lipids, complex lipids, or fats and triglycerides. And well, actually there's four, because there's phospholipids, that's different. And then the other group is the steroids. A steroid is four interconnected rigid carbon rings characterizing this group. And there's a 1, 2, 3, 4 carbon rings. If the steroid has an OH group attached right there, we then call it a sterol. Sterols are an important part of the animal plasma membrane, where we have cholesterol, which is a sterol. In fact, that's shown here. It's inside the cell membrane. So we do have phospholipid molecules in our membrane, and it is the main molecule, but we also have cholesterol. And the purpose for cholesterol is to give strength to the cell membrane. If you remember, animal cells don't have a cell wall, and so they have cholesterol in their cell membranes to give them a little more strength. Cholesterol is only found in animal tissue, meaning in animal cells. It is not found in plant cells. Any question about any of that? Steroids are a lipid which is not made up of a monomer. A steroid is one molecule, like cholesterol right here, and it has no monomers. You wouldn't call it a polymer either. It's a large molecule, but it's not a polymer because it's not made up of smaller subunits. All right, any question about lipids? If not, let's move on to proteins. Proteins are essential in the cell structure and function of both a cell and an organism. Proteins differ. They may be structural proteins found in bone, muscle, or hair. They may be enzymes which speed up chemical reactions. They may be transport proteins that move chemicals across the cell membrane or across a organelle membrane. They may be regulatory proteins that regulate a body function, like a hormone. Like in children, they have a growth hormone. Proteins may be responsible for movement, and it's true that we have proteins in our arms and legs for moving, but you also have proteins for cellular movement, such as the protein that makes up flagella. Proteins can be responsible for some bacterial toxins. So the protein itself is toxic. And then proteins can be antibodies, proteins that help defend an organism against an infection by a microbe. So this tells you that proteins have a bunch of different functions. Proteins are more diverse than any of the other four biological molecules. They are more diverse than the other biological molecules because they aren't really necessarily larger, but they have more building blocks, like complex carbohydrates only have a handful of building blocks, such as glucose, fructose, and galactose. And fats like triglyceride or lipids only have a few building blocks, and some of them, like cholesterol, have no building blocks. Cholesterol is just one molecule. But like a phospholipid has three building blocks, a fatty acid, a glycerol, and a phosphate group. See, carbohydrates have only the simple sugars, which are a handful of building blocks. Oh, I was trying to remember the other group. The nucleic acids only have four building blocks, the different four nucleotides. But proteins have a building block that we call an amino acid, and there are 20 different amino acids, at least in humans, and in most organisms, they have 20 different amino acids. So proteins consist of the monomer that we call amino acids. This is the generalized shape of an amino acid, where it has an amino group on one end, or carboxyl group on the other end. Between them, they have the central carbon. Hydrogen is connected to that central carbon. And then on the fourth position of the carbon, we have an R group. The R group can vary. R stands for, well, essentially anything we want it to, but in amino acids, it's the R group that varies that makes the different amino acid. And then you just link these amino acids together to form a string of them to make a protein. Here we're looking at one R group for tyrosine. Here are some of the different R groups. You don't need to know these R groups. I'm just trying to show you that each amino acid will have a different R group. So there are 20 different amino acids. That means there are 20 different, at least in humans, there's 20 different amino acids. So there will be 20 different R groups, one for each amino acid. The simplest R group is simply hydrogen, which we see on glycine right here. Your R group can be, it can be nonpolar, like a methyl group here, a branch methyl group here, a branch chain of methyl groups there. These are all nonpolar R groups. But the R groups don't have to be all nonpolar. Some R groups are, or at least have regions that are polar. Like this R group here has this OH group, and that's polar. The rest of the R group is nonpolar. In this R group, wherever there's the NH, it is polar. And then this region of the R group would be nonpolar. Better shown on this one. So this would be a polar region of the R group, and then the rest of the R group would be nonpolar. Amino acids always have a polar group. Actually, they have two polar groups because the amino group is always polar. The carboxyl group is always polar. And then the R group can be either nonpolar like that one, or it can have polar regions like this one. Any question about that? Okay, amino acids are linked together by a peptide bond. A peptide bond is formed by dehydration synthesis. You remove the OH from this amino acid, the hydrogen from that amino acid, and then that combines the nitrogen with this carbon here, which is an unusual unusual bond between nitrogen and carbon. That's not only unusual in living organisms, it's also unusual in the universe. We don't often have a bond between nitrogen and carbon. We do have it in amino acids and proteins when they the amino acids link together. And so we call this bond a peptide bond because it is found in peptides. And if you remember, a peptide is a shorter version of a protein. When you're adding amino acids together, we call it a peptide until you add the last amino acid on, in which case we then call it a protein. So peptide bonds are formed by dehydration synthesis, and we link the amino acids together to make a polypeptide chain. And when the last amino acid is joined to the growing protein, we then call it a protein. But it's not a protein until the last amino acid is added. You can call it a growing protein, but not a protein. There are 4 levels to the protein structure: a primary level, a secondary level, a tertiary structure or level, and then a quaternary level or structure. The primary, well, let me go ahead and show you the movie.
The hardest part about a writing assignment is getting started. I'll use Grammarly to illustrate how AI can be your...
In this
Okay,
video, we're going
commercial.
to talk about the structure of proteins. And what basically is a protein? A protein is a polymer consistent of many amino acids. So each amino acid represented by the circle is a monomer that forms the protein. Whenever you have many amino acids, it's also called a polypeptide. And the bond that connects each individual amino acid residue is a peptide bond. So let's talk about the individual structure of an amino acid. So an amino acid has a chiral carbon with a hydrogen attached to it, and it has an amine group attached to it as well. And then it has an R group and a carboxyl group. Now I'm going to draw another amino acid right next to it. The left side, where the nitrogen is located, is the N-terminal of the amino acid. The right side, that has the carboxyl group, is the C-terminal of the amino acid. Now these two amino acids can react with each other in a condensation reaction where we're going to lose water, and we're going to connect the two amino acids into one molecule. So once water is removed, we could form a peptide bond. This is also called a dehydration reaction because you're losing water. And so this is going to be the new product of the reaction. So let's see if I can fit it in here. So this oxygen is gone. So now we have a nitrogen. Let me take out H2O. We have one hydrogen left over. We lost 2 once we lost water. So this is a dipeptide because we have 2 amino acids combined. And so this amide bond that we see here, that is the peptide bond. we formed a bond between the carbon and the nitrogen atom. So whenever you combine two amino acids to form a peptide bond, you're forming a covalent bond, which is hard to break. Now let's talk about the different levels of protein structure. There's 4 levels that you need to know: the primary structure, the secondary structure, the tertiary structure, and finally the quaternary structure. Now the primary structure is simply based on the sequence of the amino acids found in the protein. The sequence determines the shape of the protein. If you replace just one amino acid with another, it will completely change the shape of the protein. So the shape and its function is primarily determined by the sequence of amino acids. The secondary structure describes the localized shape of a protein. And there's 2 of them that you need to be familiar with: the alpha helix and the beta pleated sheet. In the alpha helix, you could see that it's stabilized by hydrogen bonds. The NH group of one amino acid interacts with the carbonyl group of another amino acid. And each turn contains about 3.6 amino acid residues. Now here is a visual illustration of the beta pleated sheet. Just like the alpha helix, it too is stabilized by hydrogen bonds between the carbonyl group of one amino acid and the NH group of another amino acid. Next up, we have the tertiary structure, which represents the 3-dimensional complete folding pattern of the protein. And so here's a visual illustration of it. So we could see some areas is just a straight chain. Here we have an alpha helix, and here we have a beta pleated sheet. Now, the tertiary structure is one individual subunit. When you combine multiple subunits, you create a quaternary structure. So hemoglobin is an example of that. It has 4 individual subunits, 2 alpha subunits and 2 beta subunits. So hemoglobin is a protein with a quaternary structure. And so that's basically it for this video. Hopefully it gave you a basic understanding of proteins and the 4 levels of structure that they have. Thanks for watching.
All right, any questions about that video? So let's talk about the 4 levels of protein structure. The primary structure is the specific sequence of amino acids in a polypeptide chain. So when the protein is made, it will simply be a sequence of amino acids, one after the other. And that's the primary structure. Any question about that? Now, when the protein is first made, it's just a linear sequence of amino acids. However, the secondary structure soon forms as the amino acids of the protein start interacting with each other. And initially, it's only localized interactions where neighboring amino acids interact with each other. And that we call the secondary structure. There are different types of secondary structure. The video told you there are 2 you need to talk about. There's actually 3. You don't really need to know these, but you can have the alpha helix, which is a shape like that or like that. The beta pleated sheet, which is a shape like this. And then there's the random coil, and let me just go ahead and draw out the random coil. Try and find paint. The random coil is where the amino acid the protein just randomly takes on a shape. Let's make that come out a little further. Something like that. The secondary structure, where do I have that? The secondary structure happens by localized interaction where the different neighboring amino acids start reacting with each other. And I think your textbook and many internet sites say that the secondary structure only happens by hydrogen bonds. That isn't correct. It is true that hydrogen bonds are the primary way the secondary structure happens. However, there are other interactions that can cause the secondary structure. Let me blow this up. So in the secondary structure, local amino acids can interact with each other. These two can be attracted to each other, and that could be from a polar molecule being attracted to a polar molecule, and they can form a hydrogen bond between them. However, these two can form an ionic bond between them, and this happens when an R group loses a hydrogen, or you can have the amino group. Let me see which one loses it. The carboxyl group loses the hydrogen, and then the amino group of this amino acid gains the hydrogen. That will make one positively charged and one negatively charged. And if the hydrogen moves from this one to that one, this one will be negatively charged and that one will be positively charged. That will make an electric attraction between the two charges, and that will form an ionic bond between this amino acid and that amino acid. So those are the two attractions. There's one other attraction. Let's go ahead and make it this one here. Let's say these two amino acids are attracted to each other. This can be a van der Waals forces, where this is a non-polar, come on mouse, a non-polar amino acid, and that's a non-polar amino acid. They have non-polar R groups, and they can be attracted to each other because non-polar molecules like to be together. And if you think about it, when you have a bunch of oil molecules like your corn oil or your olive oil in your container, the molecules have to be attracted to each other to be a liquid. Because if the molecules weren't attracted to each other, they would not be a liquid, they would be a gas. And obviously olive oil and corn oil is a liquid. And with olive oil, it's very easy to see that there has to be some attraction between the molecules, because if you put the olive oil in the refrigerator, the oil will no longer be a liquid, it'll be a solid. And what you've done is you've changed the attraction between the molecules so that the temperature is such that the temperature can no longer break the attraction between those two molecules. And that's why olive oil will become a solid in the refrigerator. Anyone ever do that? Put olive oil in the refrigerator? Nobody's going to admit, well, I'll tell you, I've done it. And like I said, there is attraction between the molecules, and that's why oil, olive oil or corn oil, is a liquid. Because if there was no attraction, it wouldn't be a liquid, it'd be a gas. And then if there's total attraction, meaning the molecules can't break the attraction bond between them, it would be a solid. And you can, like I said, demonstrate that by putting the olive oil in the refrigerator. My point is there is attraction between nonpolar molecules, and we're not going to talk much about it. It's going to be called van der Waals forces between the two molecules. It's much weaker than a hydrogen bond, but there is attraction between them. There can also be repulsion between two amino acids, like this amino acid. Come on, my mouse isn't working. This amino acid is being repulsed from this amino acid. This can be a non-polar amino acid. This can be a polar amino acid. These two will then be repulsed from each other. The other way they can be repulsed is if this has a positive charge, and that is a positive charge, then these would be, excuse me, these would be, I got the hiccups here. These would be repulsed from each other, meaning they will not want to come together. They'll separate from each other. My mouse keeps dying. So those are the forces that can cause the secondary structure, the attraction, and the repulsion between neighboring amino acid. You only find the secondary structure between neighboring amino acids. Any question about the secondary structure? The secondary structure does give rise to shape in the protein. Instead of being a simple straight line of amino acids, the amino acids will take a shape where there will be attraction. So these two will come together, those two will come together. These are repulsion, so they'll be away from each other. And that's the alpha helix. The beta plated sheet, it has attraction between one amino acid and another to make the beta plated sheet. And usually that attraction is hydrogen bonds. The random coil, let me show you that again. The random coil has attraction between nearby amino acids. So I don't seem to have the. So this amino acid will be attracted to this amino acid. This would be repulsed. Well, let's go repulsed here and repulsed here. That's why they're not together. And then these would be attracted to each other. We'll just go like that. Because there's amino acid right here and amino acid right there. They're attracted to each other, and that's why they're lining up right there. And I guess you'd say these, let me make that a little closer. So these would be attracted to each other right there too. That would probably be repulsed from here to here, and obviously repulsion from this one, and so let's say that one. Any question about any of that? All right. The tertiary structure is interaction between amino acids, but they do not need to be neighbors. The tertiary structure can have amino acids interacting with each other all over the protein. For example, right here, we have attraction between this amino acid and this amino acid. This amino acid is at the end of the protein, and this one is near the beginning of the protein. For tertiary structure, it gives the protein its shape, where what used to be a single line of amino acids linked together in the primary structure and a single line of the amino acids, it gives that protein now the ability to wrap on itself and supercoil. You can say making a knot if you want. And that we call the three-dimensional structure of the polypeptide chain. If there's only one polypeptide in the protein, that gives the three-dimensional structure of the protein. For the tertiary structure, you have all of the forces of attraction and repulsion that we saw in the secondary structure. So there can be attraction between two polar amino acids. There can be attraction between two nonpolar amino acids. They can form hydrogen bonds. They can form ionic bonds. That's attraction. They can be repulsed from each other, where both amino acids have the same charge. They will be repulsed from each other. And they can be repulsed from each other if one amino acid is nonpolar and the other amino acid is polar. So we see all of those in the tertiary structure. However, there is one interaction in the tertiary structure we only see in the tertiary structure, meaning we only see it with the tertiary, not with the secondary structure. And let me go ahead and I'll just blow this up, maybe talk about it from here. And that would be the disulfide bond that can form between two cysteine amino acids. So if there's a cysteine amino acid over here and a cysteine amino acid over here, I guess I do need to show that on the paint again. So let's make this one here. This protein like that. And there's a cysteine here. And a cysteine here. I'll try and write that over here. Cysteine. All right, cysteine has a, maybe I should move that. Yep. Oh, didn't mean to do that. Cysteine has a hydrogen sulfhydryl group. Let me just put a dot there and a dot there. Group. There we go. All right, so the cysteine, or the sulfur will be connected to the cysteine. It's actually part of the cysteine. And these hydrogens can come off. Gonna erase them. And then the sulfur can form a covalent chemical bond between the two sulfur of the cysteine. This we call a disulfide bond because there's two sulfurs. This is a very strong bond, much stronger than a hydrogen bond. This is a covalent bond between these two cysteines that form. Any question about that? Anyways, you see this tertiary interaction between the two cysteines where the hydrogens come off and then the sulfur forms a covalent bond between them. And that is the only difference really between the different attractions and repulsions between the tertiary structure and the secondary structure. The second change is that the tertiary structure is a three-dimensional structure. If a protein only has one polypeptide unit to make up the protein, then the tertiary structure is the highest level of structure the protein can have. And every enzyme that I know of only has one polypeptide to make the enzyme, like catalase, catecholase. I suppose if we're talking about polymerase, there's many proteins that make up that one, but that's because it's a protein, several proteins joined together in a, what do you call that? A conglomeration of proteins that work as a unit to like synthesize the DNA. But generally one enzyme, if it's a single enzyme that only does one thing, meaning a normal enzyme, it has only one polypeptide to make up the enzyme. And then it'll only have a tertiary structure. However, some proteins have more than one polypeptide to make up the protein. And these proteins have a quaternary structure. The quaternary structure is when the different polypeptides come together to make the final protein. And all of the forces of traction that we discussed in the secondary level and then in the tertiary level can happen in the quaternary level. So for example, let's say this is an antibody or hemoglobin. It's made up of 4 different polypeptides, 1, 2, 3, 4. These polypeptides come together, and there can be attraction between this amino acid and that amino acid. There can be repulsion between this amino acid and that amino acid. And these different amino acid interactions make up the quaternary structure. You might know that hemoglobin, insulin, and antibodies have a quaternary structure because they're made up of 2 or more polypeptides. I know that hemoglobin and antibodies are made up of 4 polypeptides. Any question about any of that? All right, with a little further discussion of proteins, we have two overall shapes a protein may take. There's a globular protein, and this is a compact protein, roughly spherical. Hemoglobin is an example of a globular protein. And then the second type of proteins you can have is a fibrous protein. This protein is thread-like. We find fibrous proteins in the structural proteins like collagen, whatever makes up bone. I forgot the name of that protein. Hair, which is mainly, I think, collagen anyways, but you can also have conjugated proteins. These are a combination of a protein or amino acids with other organic and inorganic components. The proteins are named by their non-protein component, meaning a glycoprotein is a protein bound to well, it's a carbohydrate, but a glycogen, that's what I'm trying to say. Usually a sugar. You can have a lipoprotein. This is a protein combined with a lipid. And these are just different ways of having more complicated proteins. Have the protein bind to another molecule. Any question about any of that? All right. Proteins can go through denaturation. Denaturation is when the protein loses its three-dimensional shape. That can happen by breaking hydrogen bonds, but it can also happen where you go through denaturation when you form hydrogen bonds that shouldn't be there. For example, if you put a protein that should be at 37 degrees and you put it in the refrigerator or in the freezer, you can cause hydrogen bonds to form in that protein, and that these hydrogen bonds shouldn't be there. When the protein is totally denatured, the protein will no longer be functional. And then we say the protein is denatured. You can denature a protein by putting it at the wrong temperature, the wrong pH, and the wrong salt concentration. An example of a denatured protein is when you crack an egg and the egg white comes out. That liquid egg white is mostly albumin, and that protein is not denatured. You then cook the egg and you get the egg white. That egg white is very different than the liquid egg white from the egg, meaning it's no longer a liquid, it's a solid and it's white. And that's because the protein has changed its three-dimensional shape, being denatured by heat. Any question about any of that? All right, let's talk about the fourth of the biological molecules, nucleic acids. There are two nucleic acids you need to know. There's DNA, also called deoxyribonucleic acid, and RNA, or ribonucleic acid. All nucleic acids, meaning DNA and RNA, consist of a monomer, and this subunit that's the monomer we call a nucleotide. A nucleotide itself consists of three parts. A 5-pentose sugar, sorry, shown right here, a phosphate group shown right here, and then a nitrogen-containing base shown here. And there's the nucleotide. Any question about any of that? Oh, here we go. I'm saying, what's the difference between these two slides? The nitrogen-containing base can be a purine or a pyrimidine. Now, where do I want to talk about that? Oh, here we go. The purines are adenine or A and guanine or G. And I don't care how you know the names. You can know them adenine, guanine, cytosine, or thymine in DNA, or you can just call it A, G, C, and T. These are the bases. The pyrimidines are C and T, or cytosine and thymine in DNA. How you can remember which one is which is the word pyrimidine has a Y in it. All pyrimidines, I shouldn't say that, all pyrimidines in DNA have a Y in it. So cytosine has a Y in it, thymine has a Y in it, and cytosine and thymine are pyrimidines. Purines do not have a Y in them. Adenine and guanine do not have Ys in them. Any question about that? All right, the nucleotide bases bind to each other via hydrogen bonding. A and T are complementary bases, and the A will bind to the T in the other strand of DNA, meaning the A in one strand binds to the T in the other strand of DNA. This is a hydrogen bond. And then the C and the G are also complementary bases. The C of one strand of DNA will bond, hydrogen bond, to the G in the other strand of DNA. Hydrogen bonding is shown here. Let me blow that up. Where adenine here is binding to T, the AT hydrogen bond is actually two hydrogen bonds between the two nucleotides. The CG, do I have that there? Well, kind of. Shown here, let me blow that up. The A and the T have two hydrogen bonds between them. The C and the G have three hydrogen bonds between them. So the C and the G is a little stronger connection than the A and the T. Now remember, this binding of one strand of DNA to the other strand of DNA is only hydrogen bonds. Hydrogen bonds are very weak, and this bond right here, the top one, will break and reform all the time. And that's because hydrogen bonds are very weak, and at least our body temperature, 35 degrees and even 25 degrees, that hydrogen bond is weak and it will break and reform. How the two strands of DNA are held together when this hydrogen bond breaks is all the other hydrogen bonds are holding the DNA together. All right. DNA has the sugar deoxyribose. DNA is a double-stranded molecule. So normally DNA is a double-stranded molecule. Now it doesn't have to be, and in fact, our DNA is single-stranded whenever the DNA is being transcribed to make a RNA in transcription. So that's one time our DNA is single-stranded. And the second time DNA is single-stranded is when the DNA is being replicated. There will only be one single strand of DNA. The sugar phosphate backbone is what holds the single strand of DNA together. And this backbone is a chemical covalent bond. So the single strand of DNA is held together by a covalent bond. The two strands of DNA, the double strand, is held together only by hydrogen bonds. Hydrogen bonds are much weaker than a chemical bond. The covalent, not covalent, the losing my mind here. Let's see. They have an ionic bond and a covalent bond. So the covalent bond holds a single strand together, and it will hold together even if you were to put this DNA in boiling water. If you put the double strand DNA in boiling water, it would soon become single strand DNA because the boiling water would break all of the hydrogen bonds. And you don't even need to get to boiling water and break all of the hydrogen bonds. It's something like about 71 degrees will break all of the hydrogen bonds and make the DNA single-stranded. The bases are not directly in the single-stranded DNA. They do not hold the single-stranded DNA together. The bases are off from the sugar phosphate backbone of the single-strand DNA, and they make the steps for DNA. So the DNA literally looks like a spiral staircase with the two nucleotide bases facing each other and forming hydrogen bonds between them. So DNA actually looks like a twisted ladder, and it's the nucleotide bases which make the rungs of the ladder. Any question about any of that? All right, let's talk a little bit about RNA. RNA has the sugar ribose. RNA is only a single-stranded molecule, at least in our cells. Normally, RNA is a single-stranded molecule, and in our cells, we essentially only see single-strand RNA molecules unless you have a viral infection. And we'll talk about that when we talk about viruses. Once again, it's the sugar phosphate backbone which holds the RNA together. And that sugar phosphate backbone forms a covalent bond between one nucleotide and the other to make the single strand of DNA. RNA also has 4 bases. However, only 3 of the bases are the same as DNA. Where do I have that? Here we go. So in RNA, we have adenine, guanine, and cytosine, and all of these we can find in DNA. DNA also has thymine. We do not find thymine in RNA. Instead, we have uracil. Any question about any of that? So there are 4 bases to make up RNA. There are 3 major kinds of RNA. in cells. There's transfer RNA, also called tRNA. There's messenger RNA, also called messenger RNA. And there's ribosomal RNA or rRNA. Any question about any of that? All right, here's a question for you. What's the difference between tRNA, messenger RNA, and rRNA? What is the difference between those three forms of RNA? Obviously, they have different function. I'm trying to say, what is the difference to make each of them a tRNA or a messenger RNA or an rRNA? Anyone have a guess? No guess?
Their functions and structure.
What's different in their structure? They obviously have different functions, but what's different in their structure? Do they have the same sugar? Yes, they have ribose. I'll answer it since no one's answering it. Are they all single-stranded? Yes, it's single-stranded. Do they have the same 4 nucleotide bases?
No, isn't it the uracil base?
No, uracil is in RNA. Thymine is in DNA. And those are different bases. Okay. However, between tRNA, mRNA, and rRNA, they have the same RNA bases. So there's no difference in the nucleotide basis of tRNA, messenger RNA, and rRNA. So how do these molecules differ then?
Are their amino acids different?
It's not amino acids, but you're on the right track. And how they differ is the sequence of nucleotides. If the sequence of nucleotides is a tRNA, then that RNA will be a tRNA. And if the sequence of nucleotides is an rRNA, then it will be, the RNA will be an rRNA. Messenger RNA is the most different sequence because there's many different messenger RNAs, but a messenger RNA has a specific sequence too. And so that's the only difference between the different types of RNA, and that is the sequence of the RNA. Is that clear to everyone? Anyone confused on that? I'll ask you another question, like, how do you know, how does a gene or a polymerase molecule know which RNA to make? But I'll ask that question later in the term. All right, another molecule which falls under the category of a nucleotide or a nucleic acid is ATP. It is a nucleotide, by the way. It is adenosine triphosphate. We need to talk a little bit about ATP because it is the primary energy source for all cellular activities. Whenever a cell needs energy, it tends to get that energy from ATP. ATP is simply a nucleotide containing 3 phosphate groups. So it has adenine over here, the nucleotide base adenine, and then it has ribose right here, and then the 3 phosphates here. ATP has a high energy bond between the third and the second phosphates, so that this chemical bond right here is a high energy bond. And that's why it's shown by the squiggly line, because it's a high energy bond. The second phosphate, from the second phosphate to the first phosphate, also has a high energy bond. And that's right there. If the cell needs energy, it typically will break this high energy bond, and then that will release a lot of energy, and the cell will use that energy for its energy needs. Like, for example, if the cell is making a bigger carbohydrate, it will break ATP to get the energy the cell needs to combine to smaller molecules to make the larger carbohydrate molecule. All right, any questions about ATP? So ATP is made by dehydration synthesis, and it is broken by hydrolysis. To break the ATP, you simply break the third phosphate group by adding water, and then you'll get ADP, which is adenosine diphosphate with two phosphates. You'll get a phosphate, which is an inorganic molecule coming off right there. And then when you break that bond, you'll release a lot of energy, and the cell will use that energy for its energy needs. And so that's going this way, undergoing ATP hydrolysis to release energy. You can make ATP by ATP by dehydration synthesis by combining ADP, adenosine diphosphate, with the inorganic phosphate molecule and a bunch of energy. That will generate water and then ATP. So remember, ATP is what the cells use when they need energy. We can call ATP the energy currency in cells. Whenever you go to a store and you buy something you need, you exchange currency for what you're buying at the store, and the cell will use ATP for its energy currency. Any question about any of that? All right. So ATP is made all the time from ADP and phosphate and energy. And then we can use this ATP when we need energy. We break down ATP to make ADP phosphate, and then that releases energy. All right, let's talk just one slide about hydrogen bonds. What molecules have we discussed today and maybe last time that have hydrogen bonds that give the molecule important properties?
Water.
Water is a molecule that has hydrogen bonds that give the water important properties. What's another molecule that we've discussed today, which has hydrogen
Proteins?
bonds? Proteins, yes. DNA, another one. And what would
Amino
happen?
acids. Oh.
Amino acids, yes. What would happen to these molecules if the hydrogen bonds were to all disappear? Would you have a double-strand DNA molecule if all the hydrogen bonds were to disappear?
No,
No.
their
No,
structure would
it would
fall
not.
apart.
Yeah, and then the protein structure would obviously fall apart. So you get denaturation of the protein. My important point is that we need these hydrogen bonds for life to continue. And if we didn't have these hydrogen bonds, we would be in real trouble. All right, any question about chapter 2, I mean, chemistry? All right, if there's no questions, I'll see some of you in the at 7.
Thank you.
Thank you.
Thank you.
Thank you.